start presenting, I may not be able to admit more participants. Can somebody put on WhatsApp group that join immediately? Afterwards, I'll not admit anyone. कोणीतरी जरा WhatsApp वरती टाका ना. माझा फोन चार्जिंगला आहे. Because there are only 21 students which I can see over here. I think we are waiting for almost 10 minutes. So if you have seen the PowerPoint, you must have realized what are the basic characteristics of Annelida, and we will see one by one what are the general characters. Now regarding the questions, there are two types of questions which can be asked during your examination. That is, describe the characteristic features of Phylum Annelida, or what are the salient features of Phylum Annelida. This can be one question, a full question, and there can be describe the classification or different classes which are under Phylum Annelida. For all the animal classification units which we are studying, this pattern of the question paper will be there. That is, either describe the salient features or characteristic features of one particular phylum, and then coming to the classification. To understand what are the main classes under that, and then you can probably describe all those classes with their typical features and the exam. I'll admit someone is there, because what happens when I start presenting screen, it is very difficult for me to get interrupted admitting the students. I'll wait for 2 more minutes, otherwise I'll start presenting my screen. There are 22 students, so I don't think other students are going to join. I don't know what is the reason behind that. Maybe internet issue for so many people. I think 33 or 34 students are admitted to TYBSc Zoology, but only 23 are there. So 10 students have still not joined. Can you all see my screen?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay. Because like you, this is also Zoom, and using all these platforms, there might be certain troubles. So I think nobody else will join. We'll start with our discussion today. So as we all discuss, and you all have decided that we will start with animal classification, we will begin with my unit, that is unit number 3 from this paper, and this begins with Phylum Annelida. So when we discuss the characteristics of phylum, there might be certain terms which may be new to you, because those terms or explanation for those terms will be covered in first unit. Like when we say that these organisms are coelomate, you might wonder what coelomate is. So if at all you have all these questions, then I request all of you to put all these questions in the Google Classroom. Wherever I have uploaded this PowerPoint, you can post your opinions, you can post your questions in that classroom just below where I have put my PowerPoint. So you type your questions over there, classroom chat, and I'll answer for all those questions. So let us see what are the general characters of Annelida. These are the organisms which in first time in animal kingdom, these are the organisms which showed true coelom. Okay, so when we talk about coelom and we talk about triploblastic nature of organisms, you have to remember that all three layers, that is ectoderm, endoderm and mesoderm, are represented in these organisms. So if ectoderm, endoderm and true mesoderm is seen, then only the organisms will be called as triploblastic organisms. Okay, so what is a true coelom? Coelom is a body cavity and it will be called as true coelom if it is having lining of mesoderm. So first time in the animal kingdom, when you start from protozoa, then porifera, cylindrata, when you reach up to Annelida, first time in animal kingdom, you will find that annelids are the organisms or the worm-like organisms which represent true coelom, triploblastic, and they also show segmentation. Now, what is metameric segmentation? The body is divided into smaller regions or smaller parts which are called as segments. Now, there might be set of certain structures, certain features which will be repeated in every unit. In every segment, you will find a set of structures and it will be repeated for all over the entire body. So this is called as metameric segmentation. So when you talk about Annelida, this is the first important character, that is presence of true coelom, triploblastic nature representing ectoderm, endoderm and mesoderm, and presence of metameric segmentation. These three are important characters of Annelida because in all lower organisms, you do not come across with these characters. Now talking about habit and habitat, where do you find these organisms? Whenever we talk about Annelida, you have to always remember the three important representatives of this phylum, three important organisms from this phylum, that is Nereis, earthworm and leech. When you are going to describe all the characters, remember your description will go along with these three organisms in your mind, that is Nereis, earthworm and leech. So with reference to these three organisms, you can remember what is the habit and habitat for all annelid organisms. They can be aquatic, like leeches, you find them in water, aquatic organisms, or terrestrial organisms or burrowing organisms. If terrestrial, they can be of burrowing nature. Now, you might be familiar with leech and earthworm, okay? But you might not be familiar with the third organism which I mentioned, that is the Nereis. Now, Nereis is also in appearance, it is similar to earthworm, but Nereis, you find it in mud flats, especially near estuaries and creeks. Whenever we talk about intertidal region, भरतीचं पाणी येतं आणि मग नंतर ओहोटीला ते पाणी कमी कमी होत जातं, तर याच्यामध्ये जो एरिया असतो दॅट इज कॉल्ड ॲज इंटरटायडल रिजन. So intertidal region, whenever it is made up of mud flat, you find Nereis are the worm-like organisms present in this mud flat. Okay, so you can find the organisms in aquatic, terrestrial, or they might be of burrowing nature like that of earthworm. Now, what is the shape of body? The body is of tuberculous nature, that is tube-like shape is there, it is elongated. When we say worm-like or tube-like, it is elongated body, and the entire body is covered with a cuticle. Now, what is a cuticle? It is a body layer, something like a body wall, but it is the extra layer which is present along the body wall, which is of thin nature. It is not very tough. When you go to insects and other organisms, or maybe worms like ascaris, nematode worms, they are also having cuticle, but which is of tough nature because those are parasitic worms. These are worm-like organisms. Annelids are worm-like organisms, and body is covered with a cuticle, but this cuticle is very thin and very soft because many of the organisms, they use their integument or body wall as the organelle for respiration. So if the cuticle is not soft or if it is of very tough nature, then the exchange of gases will not be possible, and hence the presence of thin and soft cuticle, it is characteristic of annelid worms. So the next character is presence of true coelom. What is a true coelom? It is a body cavity. So the empty space, the space between body wall and the digestive system, this is the coelom which is lined by mesoderm, and in case of annelid worms, this coelom gets filled with fluid which is called as coelomic fluid. This is the fluid which is present in coelom, and hence it is called as coelomic fluid. Coming to symmetry, There are different types of symmetry which will be discussed in unit 1. And the three types of symmetry, that is bilateral symmetry, radial symmetry, or asymmetric organisms like that of amoeba. So annelid worms, they exhibit bilateral symmetry. If you draw a vertical line in that diagram of any annelid worm, you will find that left hand side is image of right hand side. So this is a bilaterally symmetrical organism. Body plan is tube within the tube. You can see a tuberculous nature of the body in which the entire digestive system will be placed. So this is called as tube within tube body plan. Then the body cavity is divided into many small regions or many small chambers. And why this division is possible? It is possible because of thin fiber-like structures which are known as septa. So if you open the organism, I have shared virtual dissection of earthworm in Google Classroom. If you see that dissection, you will follow that if you open the annelid worm like earthworm, when you give a cut along the body and open the body wall, you will find white tissue like things, fiber like things. These are nothing but the septa. So because of presence of this septa, the body is divided into smaller chambers. This is one example which is known as sabella. I thought of adding this photograph over here because we are mostly familiar with Nereis, earthworm and leech. But there are some beautiful annelid worms, especially if you get opportunity to visit beaches or certain marine areas, you will find beautiful tuberculous worms over there. And what happens? These are mostly tube dwelling organisms. I'll move to the next slide. You can see over here on left side you have earthworm, right side you have Chaetopterus. So here you can see the Chaetopterus has formed a tube. The brown, light brown color is the mud inside which the worm is present. You can see the entire body is present inside the mud. There is a tube-like structure which is formed and the worm is staying inside that tube. And hence these organisms, they are known as tube-dwelling organisms. I remember we had visited Narara before few years. We had been to our excursion in Gujarat coast, and Narara is a beautiful wildlife sanctuary over there. So, and it is near Jamnagar. So when we were walking on that beach, many a times we could see this Sabella. I'll again go to Sabella. Sorry, I'll again go to Sabella. So we could see while walking that sometimes through small pores, these beautiful organisms, they just peeped out. We could see the feather-like structures, very beautifully colored. And if you start walking towards them, they stage themselves inside. Okay, so tube-dwelling organisms which are present on marine substratum, marine region. So these are the examples: Sabella, then earthworm, Chaetopterus. Before few years, we had Chaetopterus specimen in our lab museum, but now because of preservation problem, we don't have the entire intact specimen. These are very delicate organisms, soft-bodied organisms. Now coming to the character, that is presence of true coelom and triploblastic, you can see the section, you can see the yellow colored central part, that is the digestive tract, surrounding which you can see the tissue layer, okay, lining of coelom. And from mesoderm, the internal organs are suspended. The empty white spaces, these are actually the coelom parts which are seen in this diagram. You can see blue colored body covering, that is ectoderm, and then digestive tract, that is from endoderm, and in between whatever is there, it is the mesoderm. So you have to remember the organism is triploblastic in nature, ectoderm, endoderm, and in between mesoderm is there, and the presence of true coelom is rightly represented in this diagram. Now coming to the locomotion, see whenever you are describing any phylum, the general characters or special features of any phylum, you have to deal with habit and habitat, that is where you get the organisms, where you find these organisms, what is the symmetry, whether the coelom is present or not, whether the organism is diploblastic or triploblastic. Then you come to locomotion, that is there any locomotion, and if it is there, how it is made possible. Then you can discuss respiration, circulation, digestion, excretion, and finally you come to nervous system and reproduction. So please remember, when you are describing characteristic features for any phylum, you have to deal with all these main points. Okay. Now coming to locomotion, please observe this diagram carefully. On left hand side, you can see that there are certain lobe like structures. Now what are these lobes? They are fleshy in nature. Okay, the lobes are fleshy in nature, and they are called as parapodia. Okay, now on this parapodia you can see that in first diagram on left hand side you can see the lobes are further divided into notopodium and neuropodium. So each parapodium, it is divided into notopodium and neuropodium, and on that you will find tuft of cilia. Okay, or tough hair like structures will be present. Tough, what is bristle? Bristle is something like hair. They are also known as setae. Okay, so we discussed that body is segmented. If you open the organism, you will find that there are small segments or small chambers or regions present. Now in every segment on both the sides you will find presence of bristles or setae or parapodia. I have said setae or parapodia because parapodia are fleshy structures represented in each and every segment, but these are not present in all annelid worms. Okay, there might be different locomotory structures present in different annelid worms. So parapodia are seen in Nereis. Okay, whereas setae are very well seen in earthworm. So different annelid worms, they might have different structures which are used for locomotion. Okay, parapodia are fleshy structures on which tuft of bristles will be present, whereas setae, they are very well represented in case of earthworm. Okay, so locomotion is possible because of setae or parapodia, or there might be muscles which are present. The muscle cells are used for locomotion. The muscle cells are used for wriggling movement. Okay, so they can stretch themselves over a substratum because of that muscular action. Now you can see this diagram, very interesting diagram, where the locomotion is possible, okay, without any spatial structure. Okay, read the statement carefully. Locomotion, spatial structures may be absent. There can be presence of bristles, there can be presence of setae, there can be presence of parapodia, or there may not be any such structure present. And the example is leech. Now the diagram represents how a leech moves. You please remember that leech is having presence of suckers. Not all annelid worms have this characteristic, but this is a particular class. We'll see further classification. So class Hirudina, here the organisms, they are parasitic in nature, and whenever there is ectoparasitism, then the organisms which exhibit ectoparasitism, they are having special characters because many a times they have to attach themselves to the body of host. So in case of leech, there is presence of sucker. There are anterior suckers and posterior suckers. That is, at both ends of the body, there are specialized suckers which are present. And because of sucker, the leech can take hold of the substratum, and it might be the body wall or the body of the host. I all must have heard about this. That is leech. In Marathi, it is known as Zaru. It sucks the blood. It is ectoparasite of cattle and even human being, especially in this rainy season. If there is any stagnant water or flood is present, and if you walk through such forested area, you might get uh, uh, these leeches. You might get attacked by these leeches. So see the movement carefully, that with help of suckers it will move its body, it will crawl, and it happens in really fast way. Okay, then coming to the next, that is respiration and circulation. So what are the respiratory structures? We discussed that there is presence of soft and thin cuticle, and it is kept as thin and soft because the body surface permits the diffusion of gases. I think in SYBSC also you have learned nutrition, respiration, circulation, which probably Rajkumari madam has taught you. There also you have seen earthworm as example, where the body surface is used for exchange of respiratory gases. Okay, or there in aquatic organisms, in aquatic annelids, there might be presence of gills, or head might be provided with some special structures which will be helpful during the respiration. We talked about tube dwelling annelids. So if the entire organism is present in the tube and the tube is present in the mud, then only the head will be taken out whenever there is gaseous exchange is to be done. So head might be provided with some specialized structure. Respiratory pigment, it may be of different types. It is called as crocoorins or hemerythrin or the usual hemoglobin, that is a red color. Vascular system, that is how to circulate the body fluid, it is very well developed. There might be presence of hearts, but you don't have to compare these hearts with the typical mammalian heart. These are very simple pumping large tube like structures, and hence they are called as hearts. There are dorsal and ventral vessels, and there will be a network of smaller vessels, but definitely it is a closed type of circulatory system. You must have studied that there are two types of circulation: open circulation and closed circulation. In closed circulation, there is presence of closed tubes through which the body fluid will be circulated. So in annelid worms, the type of circulation which is present, it is called as closed circulation. You can observe the body along the anterior posterior axis, and here you can see on dorsal side there will be a very large prominent vessel, and on ventral side also such similar prominent vessel will be present. So dorsal and ventral vessels, they will be connected via smaller vessels. So here, this is the plan of circulatory system. You can see that I'm referring to the namings which are shown in the diagram. Okay, this is a vermiculous organism, earthworm. So you can see a tube-like body. From left-hand side, if you start observing the diagram, you will find that there is lateral esophageal vessel. Now, what is esophageal vessel? Which is present in the region where you can see the esophagus. The name of this vessel is given with reference to the digestive structure which is present in that particular segment. So, on upper side, you can see the body wall as the limiting line of this diagram. So, on upper side, you can see the body wall. Just below that, you can see a vessel which is called as dorsal vessel. Whereas on the ventral side, you can see is a large vessel which is known as ventral vessel. You can see small swelling like structures which are known as hearts. So these are called as lateral hearts. They are not present on dorsal or ventral side, but they are present on lateral side of the body. Hence they are known as lateral hearts. There is a vessel which is known as supraesophageal vessel. Okay, so the vessel which will run above the esophagus, it will be called as supraesophageal. एसोफेगस के ऊपर जो रन होती है उसे बोलते हैं supraesophageal. Whereas the vessels which run on the lateral sides of esophagus, they are known as lateral esophageal vessels. Then you can see anterior loops which are formed because of presence of heart and some lateral vessels. Okay, then there will be ventrotegumentary vessel and subneural vessel which will be along the nerve cord. I know that drawing such detailed diagram may not be possible for you at the time of exam, but now when it is time, you can practice at least the anterior part where you can show the presence of lateral hearts. Okay, so you have to give the names in reference to the segment and the digestive structures which are represented in that particular segment. You can see over here the diagram of digestive system. I request everyone again to watch the virtual dissection so that the digestive system will be very clear to you. Now this is called as complete digestive system. In digestion or nutrition, you might have studied that the digestive system can be of two types. It can be incomplete or it can be complete. So when mouth and anus both are represented, it is called as complete digestive system which runs from anterior mouth to the posterior anus. So in case of earthworm, when you open the tubular body, you will find that mouth is present in segment number 1. Now I request everyone to refer to this diagram of alimentary canal, and on left hand side you can see that every segment is numbered over here. We discussed that what is the characteristic of phylum Annelida? It is presence of metameric segmentation. So all these segments are numbered over here. You can see over here numbers running from 1 to 34. There are further segments which are present over there. Okay, but only part is shown over here. Okay, so you can see the mouth which represents the anterior most part of alimentary canal. Then there is a small cavity or chamber which is known as buccal cavity. It is followed by a bulb-like pharynx. Okay, so this is the beginning of alimentary canal. Then there is a short tube-like esophagus, and then there is gizzard. Gizzard is used for churning the material. We know what is a food for earthworm. Earthworm lives in soil. So there might be decomposing and decaying material which is taken up by earthworm, and then this material is passed through the alimentary canal, and gizzard is used for churning. Then you can see a tube like stomach, and then the entire remaining body shows presence of intestine. Okay, you can see the pre-typhlosolar part of intestine. Typhlosole is a region, a particular region or part of intestine. If you go to segment number 26, 27, 28, below that you can see typhlosolar part of intestine. Okay, so the part which is present before the typhlosolar region, it is called as pre-typhlosolar part. Okay, and then there will be typhlosolar region. Entire after typhlosolar region, you will find that intestine will run towards the posterior end, and it will end as anus. Okay, so what you have to remember is the digestive system is of complete type and digestion is extracellular. Then whatever are the excretory products of this digestive system, these are excreted out by presence of specialized structures which are known as nephridia. Now these nephridia you can see in this diagram. In every segment, remember, in each and every segment, these segments or small chambers of the body are separated due to presence of septum. Okay, the fiber like tissue like thing which is known as septum, and on each septum there will be a large number of nephridia present. Earlier when we used to have mounting and dissection for courses in zoology, we used to do this mounting in FYBSc, that is mounting of nephridium from earthworm. That is, you have to open the earthworm, take out a septum from its body which is attached to the body wall, and stain it and observe it under microscope. There you will observe that nephridia get released from this septum after slight teasing, and you can see the structure very well. Now that structure is shown over here. You can see the on left hand side, you can see that from the gut region, near the gut region, the in every near every septum there is arrangement of nephridia. You can see the nephridia arrangement in each and every segment. Okay, so it is having a loop like structure. There are three loops. Okay, first loop, second loop, and then third loop, which is also called as bladder, but this is not comparable with the bladder which is present in higher organisms. But the bladder is ending in a small opening that is called as pore. Okay, so the pore will open to the exterior of the body, and whatever are the nitrogenous waste products, those will be excreted out through this pore. So what you have to remember, what is the structure which is present for excretion? It is known as nephridia, and since they are attached to septum over here, we can call these as septal nephridia. Okay, so nephridium is the structure which is used for excretion in case of annelid worms. Now coming to the nervous system, it is very well developed. Okay, now it is called as ladder form of nervous system because there will be cords, anterior and posterior nerve cords will be present, and they will be connected to each other, and it will have a look as if it is a ladder. So it is called as ladder form of nervous system. Then we have seen the digestive system, here seen the mouth. So near that esophageal region, there is aggregation of nerve cells. Okay, something like in higher organisms, we have got a brain. The nervous system starts with brain and then it continues as a spinal cord. So over here, the structure which is comparable to the higher brain, it is called as ganglion, and due to the position of the ganglion, it is called as supraesophageal ganglion. Then there is presence of nerve cord which is double in nature, so double ventral nerve cord is present, and in every segment. Okay, in the earthworm or all the annelid worms, they are known for metameric segmentation. So in every segment, you will find that there is presence of ganglion. So ganglia, they are arranged in metameric fashion. In addition to the nervous system, there are sense organs, there are photoreceptors, there are statocysts. So some of the annelid worms, they can understand what is the difference between the pressure. Some can understand the touch because they are having tactile receptors, they are having taste buds. The power of these sense organs or the strength of this functioning of these sense organs will differ from each and every annelid worm. I'll give you some example of worms which are used for vermiculture, that is some types of earthworms. Now these earthworms, whenever there is dark, that is evening period or night, they will come out. Okay, and as soon as the sun will rise, they will go down. They will go down into the soil because they are having excellent photoreceptors. They will try to avoid the light. So what type of sense organ will be functional and much more elaborate? It will depend upon which type of annelid worm you are observing. Okay, so over here you can see the nervous system plan of typical annelid worm. Prostomium, that is the mouth. I am referring to the anterior most part of the organism. You can see prostomium, that is the mouth. Come down in segment number 3. Okay, now in segment number 3, you can observe a pair of ganglia which are known as suprapharyngeal ganglia. That is the pharynx region. If you remember the digestive system, that is the pharynx region. You can see on both sides it is having a connective. So you have to remember the pharyngeal region. There is one aggregation above the pharynx, one aggregation on uh, posterior side of pharynx. Okay, so it is suprapharyngeal ganglion and subpharyngeal ganglion, which will be connected to each other by means of connective, and then further it will continue into the nerve cord. In every segment you will find that there is a ganglion and segmented nerves are present over there. So this is a brief plan of nervous system in case of annelid worm. Now how do they reproduce? Many of these worms, they are hermaphrodite in nature, like earthworm. They will have a set of testes as well as ovaries. Okay, or in some of the annelid worms, the sexes might be separate. Coelomic epithelium, that is the epithelium, will give rise to gonads, that is testes or ovaries, and development can be direct, directly producing the worm, or it may be indirect, that is eggs will give rise to a larva. The larva may not look like the adult organism, it will, and then the larva will transform into adult organism. In last year syllabus, you have learned embryology, where you have also studied different types of cleavage. So in case of annelid worms, the cleavage is spiral and determinate. I have mentioned over here trochophore larva, which is the common name for larva of most of the annelid worms. I'll show you the diagram of trochophore larva. Sorry, I think I have missed the diagram, but I'll send you in Google Classroom. This diagram will show you circulatory, nervous, as well as reproductive system together. You can see male and female gonads over there. Okay, and red colored one are the hearts or aortic arches. On left hand side, you can see the structures of nervous system, that is the brain, and whatever is represented in red color over here, that is part of circulatory system. Okay, you can see the dorsal blood vessel, ventral blood vessel. Okay, and you can also see the nervous system over here. Now here you can see the specialized structures of reproductive system, which are known as seminal receptacles or seminal vesicles. You can see the bag-like vesicles and small bulb-like seminal receptacles. Okay, the red color ones, they are vessel-like, tube-like dorsal blood vessel is seen over here, and the green colored one, they represent the excretory system, that is the nephridium of earthworm. It is the dorsal dissection. It is from segment number 9 to segment number 15. So when you draw the diagram, you have to remember which segments you are drawing. And lumbricus, it is the genus of one earthworm. Okay, here the diagram shows how the reproduction takes place. You can see dark colored band in these earthworms. Okay, now this dark color band, it is known as clitellum. When you observe any earthworm, the common earthworm, in segment number 14, 15 and 16, there is a dark colored band which is called as clitellum. Now you can see the male pores over there, and sperm movement occurs between the two organisms. See, we mentioned that these worms can be hermaphrodite, that is male and female reproductive structures are present in the same organism. But the exchange of sperms, it will take place between two different organisms. Okay, so you can see how the reproduction is taking place and how the sperm movement will be achieved. Now, yes, this is the trochophore larva which I mentioned. So you can see apical tuft of cilia. You can see the presence of hair. The characteristic of this larva is that there are bands of cilia which are seen in the diagram. On upper side, you can see apical tuft. Now, this is the tuft of cilia, tuft of hair. Okay. And in the middle portion of the body of this larva, you can see ciliary bands. So this is the characteristic of larva. And on lower side also, you can see a bunch of hair. Okay, so here the characteristic of ciliary bands, it is seen in trophophore larva. So most of the annelid worms, after reproduction, they will give rise to eggs and which will hatch into larva, which is known as trophophore larva. This is not seen in other molluscan or higher organisms like arthropods. This is characteristic of all annelid worms. Okay, so that is the end of this PowerPoint. I hope you have followed what we have discussed. Mm, and most of the characters which are described, they are of very simple nature. That is, if you read it again and again, I think you will be very comfortable to describe. What I have shared in Google Classroom is the general characters of this phylum, plus I have also given notes about classification. I have not shared PowerPoint about classification, but I have shared e-notes. Yesterday I have sent e-notes including classification. Now I expect all of you to read those e-notes so that you will follow how phylum Annelida is classified. Now once again you have to read all the characters, and then tonight or maybe tomorrow morning I'll send you next PowerPoint which will include the classification of phylum Annelida. What we discussed, that to discuss or study any phylum, first you have to study the general characters and then you have to go to classification. So this phylum is further divided into three classes which are known as Polychaeta, Oligochaeta and Hirudina. Polychaeta, what is the example? It is Nereis. Oligochaeta, what is the example? It is earthworm. And Hirudina, what is the example? It is leech. So now we will study what makes you classify these organisms. What are the basis or what are the characters which are used to segregate these organisms into polychaete, oligochaete and hirudina? How you can identify these organisms? That will be studying in next lecture. So I'll be sharing a PowerPoint with reference to classification of Annelida. Now there is a small test which is posted in Google Classroom. I want you to go through the general characters in this PowerPoint and attempt that test. This test will be very simple of only 5 marks, but I want each and everybody to attempt it. Okay, and then only I'll come to know whether you have understood the general characters of phylum Annelida or not. I think that's all for today. You can unmute yourself if you have any difficulty. Please unmute yourself. Anyone would like to ask any difficulty, or I'll end this session over here? Any question you have to ask? You can unmute yourself. No. Okay, Mansi is saying no on behalf of all of you. So I end this session over here. You can leave the meeting and take up that test. Okay? You can leave the meeting and take the test immediately.